When we first began testing for mutations in cancers, it was relatively simple. We tested for EGFR mutations, and that was enough. We began to realize the value of KRAS mutations as well, and so we slowly added that on. And then over the last couple of years, we've heard about ALK and ROS. When you realize that you start to add more and more on, just for lung cancer, you've added 4 distinct genetic lesions over the last few years. And then you look at other diseases, so look at BRAF mutant melanoma. We want to find those patients with BRAF mutations, identify them, and give them the right treatment. So as the number of mutations goes up, it becomes clear that you can't do independent tests, especially when you realize that many of these mutations and rearrangements are rare, happening in only 1 or 2% of people. So really we're inspired by the standard laboratories we do in the clinic. When a patient goes to see a doctor, the doctor orders usually something we call a comprehensive panel. That includes some things that we really care a lot about, the creatinine, which is a good measure of kidney function. But it also contains a lot of things that are only rarely abnormal, things like chloride level and BUN, things that we care about in a peripheral sense, but we're not often ordering that blood test just for that. And I think that's the same idea when we think about multiplex mutation testing. We want to find the things that are common, but we want to also be able to find the things that are rare without going through several other steps. The several other steps waste time, and the several other steps can often waste tissue. So if you can do a multiplex test which gets all the mutations or rearrangements of interest at one time, then we're going to be able to build off that and speed things up and do a more efficient job with the tissue we have. In lung cancer, the tissue that's available is often sparse. It's getting better and better over the years as we realize this problem. But nonetheless, the number of tests we have to do quickly rises faster than we can get additional tissue. So one of the ways we can multiplex tests better is something called next-gen sequencing. Next-gen sequencing is offered by several manufacturers. Ion Torrent is one company, and Illumina produces something called the HiSeq platform. These are big machines that major institutions or companies can buy to do a variety of testing that allows multiplex testing. The amazing thing about these devices is that you can sequence multiple genes, and in the case of HiSeq, you can test for hundreds of genes, and same with Ion Torrent, hundreds of genes, find abnormalities. And these aren't looking for single hotspots like many of our previous tests. These are sequencing the entire gene. So that's an amazing thing. You can find mutations in more genes, and you can find mutations in different areas than any other device in the past. One of the challenges that brings with it is something we call computational biology. That is taking all that data that has been generated from a run off a machine and trying to sort out what is a mutation that we care about, what is noise from the machine, and what is a mutation that we don't know anything about, sorting all that out and developing a report to go back to a doctor. So that's certainly a challenge, but I think one that we're slowly beginning to meet, and a variety of companies are in this area trying to make that better for doctors and patients.